And welcome back to the podcast. Today we're stepping into one of the most extraordinary stories the human body has to tell, the story of pregnancy, growth, and development. From a single fertilized cell to a fully formed human being, development is a process shaped by precise timing, complex communication, and remarkable biological coordination. So, today we'll explore how life begins, how it grows, and how the body changes from conception through birth and beyond. So, using this chapter as our guide, we'll break down key concepts like fertilization, embryonic and fetal development, pregnancy, changes in the mother, and stages of human growth after birth. And along the way, we'll connect anatomy and physiology to real life implications in healthcare, imaging, and clinical decision making. So, whether you're studying for class, preparing for a healthcare career, or simply curious about how the human body creates and sustains life, this episode is designed to make complex processes clear, meaningful, and human. So let's go ahead and get started. Okay, so let's go ahead and begin with the overview of development. Let's start at the very beginning, literally. No, not at the asking out someone on a date, but the beginning of fertilization. The human development begins at fertilization. So that's when the sperm cell and secondary oocyte unite. And this single moment forms a zygote, a one cell structure, if you remember from last lecture, that carries the complete genetic blueprint for a new human being. So from that point forward, pregnancy has begun. And over the next 38 weeks, that single cell divides again and again. Cells multiply, specialize, and organize themselves into distinct tissues and organs. What starts out as a microscopic structure gradually becomes a complex, fully formed human being. And at the end of that process, a baby is born. But development doesn't stop at birth. So humans are constantly changing across the lifespan. So that's why you're all here in class. And in anatomy and physiology, we describe this process using two related ideas: growth and development. So growth refers specifically to an increase in size. So this happens because cells increase in number, allowing tissues and organs to become larger as the body matures. Development, on the other hand, is broader. It includes growth, but it also includes aging and all the structural and functional changes that occur during life. So development is a continuous process. It begins at fertilization and continues until death. We divide development into two major time periods: the prenatal period, which spans from fertilization to birth, when the body's basic structures are formed, then the postnatal period, which begins at birth and continues for the rest of the life as the body grows, matures, adapts, and then eventually, ages. So understanding these distinctions helps us frame everything else in this chapter because pregnancy, growth, and development, they're not isolated events from each other. They're part of this continuous human cycle. Let's continue with fertilization. So, what exactly is fertilization? All jokes aside, right? So, fertilization is also called conception. It's the moment a secondary oocyte and a sperm cell unite. And this doesn't happen just anywhere. It typically occurs in the infundibulum of the uterine, also known as the fallopian tube. But before that can happen, both cells have to get to the right place at the right time. Uh, First, the secondary oocyte must be released during ovulation and enter the uterine tube. And once released, the clock starts ticking. So that oocyte survives only about 12 to 24 hours. Meanwhile, the sperm begin a much longer journey. They travel from the vagina through the cervix, across the uterus, and into the uterine tube. And they move fast. So sperm can reach the oocyte within 1 hour of intercourse. And this is kind of like where the numbers start becoming a little bit dramatic here. So a single, we saw this video in the last lecture, that a single ejaculation contains 200 to 600 million sperm, but only a few hundred ever make it anywhere near the oocyte. The rest are filtered out along the way. Um, and the sperm that do survive, well, they have an advantage. Some of them can live up to 6 days inside the female reproductive tract, and they just wait, right? They just wait for the ovulation to occur. But in the end, it comes down to just one, right? Because there can only be one. What's the name of that movie? The Highlander. There can only be one. So only one sperm cell can successfully penetrate and fertilize the oocyte. And when that happens, the process of human development officially begins. So you have one cell, one moment, one beginning, one, I don't know. I was thinking of something poetic there. Okay, let's look at this visualization of fertilization following the cells. So if we just pause for a moment and picture what's happening, we see here in the image on the left side of the figure, you're looking at the female reproductive tract. And this is the route sperm must survive to reach the oocyte. So sperm are deposited in the vagina, but that's just the beginning. That's just the starting line. From there, they move through the cervix into the body of the uterus, and then into one of the uterine tubes. And the arrows in the figure show just how narrow and selective this pathway is. Now, if you shift your attention to the ovary, right? So during ovulation, we say a secondary oocyte is released from the ovary and swept into the infundibulum, the funnel shaped opening of the uterine tube. And this is where fertilization is most likely to occur, not in the uterus itself. So the two paths meet here. And only a small number of sperm, like I said before, ever reach the oocyte. So in the image on the right, you can see dozens of sperm surrounding a single oocyte. They're all competing, but again, only one will succeed. Kind of like ladies' night at most adult establishments. Right? And so once the sperm penetrates the oocyte, the rules change instantly. The oocyte's membrane becomes impenetrable to the others, and that ensures that fertilization happens only once. So when you look at these figures, don't just see anatomy, kind of see if you can picture the movement, see the timing, see the selection process. Again, this is not chance, it's an incredibly regulated biological process. And at this moment, right here in the uterine tube, that's where human development begins. Okay, so the sperm cell joins secondary oocyte. So again, when one sperm succeeds, let's go ahead and zoom all the way in to the exact moment fertilization happens. So when sperm reach the oocyte, they don't go straight in. First, they have to break through the what's called the corona radiata, and that's that layer of follicular cells that surrounds the oocyte. So sperm release that acrosomal um, um, enzyme, excuse me, that head, remember we mentioned the acrosome, which is the head of the sperm. They release the acrosomal enzymes, and that helps dissolve the outer barrier of the corona radiata. So next comes what's called the zona pellucida, excuse me. Um, and that's that tough glycoprotein shell tightly wrapped around the oocyte. So several hundred sperm help weaken this layer because they're all attacking it. They all want to get in, but only one's actually going to make it all the way through, and that's the rule. So only one sperm is allowed to penetrate the oocyte's membrane and complete fertilization. And the instant that sperm enters, everything changes. The zona pellucida hardens like almost instantaneously. It forms a block that prevents any additional sperm from entering. So that's why it hardens. And this is how the body ensures the correct chromosome numbers. So as the sperm nucleus moves into the oocyte, the secondary oocyte completes meiosis two, and a tiny polar body is produced and discarded while the oocyte retains the female chromosomes. Again, we mentioned all this in the previous chapter. And so at this stage, the nuclei of the sperm and the oocyte are called the pronuclei. They exist separately, and each carries half of the genetic material. And then comes that final step where the pronuclei unite. And so at that moment, fertilization is complete, and the cell is now called a zygote, which contains the 46 chromosomes. And what's that correctly? 46 chromosomes. Um, I need to enunciate my words. Yeah, chromosomes. And so it's a normal number for a human body cell. And so you got one cell, one complete genetic blueprint, and the next chapter of development begins uh, instantaneously, immediately. Okay, so let's go ahead and look at the steps of fertilization, and we're going to do like a slow motion replay as you look at this image. So let's walk through fertilization step by step. And excuse me if I really over enunciate certain words. I'm kind of practicing that I don't slur my words. Um, and so you can understand and hear me clearly. Like when I say fertilization, that's not the way anybody really says it, but I need to practice in case I ever get a job as a game show host. All right, so let's start at the outside of the oocyte. The first barrier sperm encounters is the corona radiata, if you remember that. Layers of follicular cells that surround the oocyte. Right? Sperm release the acrosomal enzymes, breaking down this protective layer, and that kind of clears a pathway forward. And just beneath that is the zona pellucida. That's that thick glycoprotein shell, right? So the sperm bind to specific receptors, triggering the acrosomal reaction. So enzymes are released, digesting like a little tiny narrow funnel through the zona pellucida, and then several sperm actually, well, they don't assist; they're all trying to get in, but in a way, it's assisting one to actually finally get in. So we say that several sperm assist in the process. They're not really assisting, but you get the idea. Everyone's kind of contributing to the ultimate, the goal, but only one reaches the oocyte's cell membrane. So when that single sperm fuses with the oocyte membrane, the membrane instantly changes. The zona pellucida then hardens. Again, we said it hardens because it prevents any other sperm from trying to make it through. So this creates a chemical and a physical block that prevents any additional sperm from entering. And again, to maintain the correct number of chromosomes. So now we're inside the oocyte, right? The sperm nucleus uh, moves inward, and this signals the secondary oocyte to complete meiosis 2. Meiosis 2. So what we do here is we're producing a second polar body and retaining the female genetic material. So at this stage, the sperm nucleus and the oocyte nucleus, they're separate. And so they're called pronuclei. Each one of them contains the 23 chromosomes. We saw these images on the last lecture. Now we're kind of going into detail of what's happening there. So finally, the pronuclei, they migrate toward each other, and then they start fusing. And this fusion completes fertilization. What ends up resulting in all of this is a zygote. The zygote. I'm gonna say it another time. Zygote. Some terms you really need to know. So that's a single cell containing the 46 chromosomes. Right. So the zygote contains the 46 chromosomes. It carries a full genetic blueprint for a human being. And from this moment forward, like we said in the script slide, I say, not the image slide here, development starts accelerating. So you see cell division begins immediately. I mean, immediately. There's no time wasted. And then the journey starts from that one cell to one body, and that's officially underway. Right? Excuse the Navy terms, the nautical terms. All right, let's go ahead and continue from science to technology. So we're going to talk about assisted reproductive technologies real quick. So um, up to this point, I've got to enunciate better. We've talked about how fertilization normally happens. So inside the uterine tube, where sperm and egg meet naturally. But biology doesn't always cooperate. There's a lot of randomness and things that can go wrong. So it's a miracle that this happens to begin with. So when there's a problem with gamete production, or when a physical blockage prevents sperm and egg from ever meeting, infertility occurs. This is one of the reasons for infertility, and that's where science steps in with technology. So these approaches are known collectively as, I'm going to say this slowly, assisted reproductive technologies, or ART. Right? So today, about 1% of births in the United States results from some sort of ART, and that may sound small, but it represents millions of families whose stories depend on these technologies. So, as an engineer, this is not my area of specialty, but there are engineers who this is all they work with. Right? We have biomechanics, bioinstrumentation people, biomaterials. So there's engineers who focus on each one of these areas. Um, anyway, that's for a whole different program, clinical engineering. Um, Spread the word. So one common approach is again donated sperm and intrauterine insemination. And so in this method, a physician places sperm directly into the cervix or uterus, and it may be used when a male partner is infertile or carries a genetic condition, or when a woman chooses to have a child without a male partner. Another widely known method is in vitro fertilization, or IVF, and this is where fertilization occurs outside the body in a laboratory setting. So once an embryo actually forms, it's placed in the woman's uterus, and IVF is often used when the uterine tubes are blocked or damaged. And we'll leave it there. We had a discussion in class about this, but I won't bring up the discussions, so this lecture can remain neutral. Uh, let's talk about a related, but again, less familiar method, and this is something called a gamete intrafallopian transfer, and it's sometimes called a GIFT, right? GIFT. And in this process, medications stimulate the ovaries to release multiple oocytes, and the largest oocytes are collected, they're mixed with sperm, and then placed together directly into the uterine tube. And this allows fertilization to occur inside the body. In some cases, this approach has higher success rates than IVF. So again, together these technologies just highlight something that's very very important because we have a specialty in medicine just for this. Um, so understanding anatomy and physiology isn't just about knowing how the body works when everything goes right. It's also about knowing how science can step in, again, ethically, carefully, and deliberately when the body doesn't. And that connection between biology and technology is where my particular specialty comes in. And that's what helps continue to shape modern reproductive care, even though reproductive care isn't my specialty of clinical engineering. So let's go ahead and look at pregnancy and the prenatal period. All right. So just so you'll know, this is section 23.3 out of your textbook. So we are on section 3 now. We've already gone through the two sections of the textbook. Section 23.3 happens to be the longest, so stick with me. Yeah, pregnancy and the prenatal period. So pregnancy is defined as the presence of a developing offspring in the uterus. So from conception to birth, the prenatal period usually lasts about 38 weeks. And during this time, a single fertilized cell develops into a fully formed human. And to make sense of this process, the prenatal period is divided into three stages. So the first one being the pre-embryonic stage, and this includes the earliest cell divisions after fertilization. Then we have the actual embryonic stage, and this is when the body's basic structures and organ systems begin to form. And then we have the third stage, which is the fetal stage. And this focuses on growth and maturation. So organs begin to develop, they support life outside the uterus, and together these three stages outline the entire journey from conception to birth. So here we're looking at the pre-embryonic stage. So the pre-embryonic stage runs from fertilization through the second week of development. This is a period of rapid change and movement. So it includes cleavage, blastocyst formation, implantation, and the early development of extraembryonic membranes. So it begins, again, like I said, with cleavage, and that is a series of rapid cell divisions where zygote cytoplasm is divided into smaller cells called blastomeres. Right? So cleavage leads to blastomeres. And the total size stays the same, but the number of cells increases quickly. Then cell division continues, and cells form a solid ball called a morula, MORULA, as shown there, and that's made up of about 16 cells, which travels into the uterus. And the morula then becomes a blastocyst, and that's a hollow ball of cells that attaches to the uterine lining. And inside that blastocyst, the inner cell mass will develop into the embryo itself. Now, the outer layer, called the trophoblast, helps support the embryo and releases HCG, the hormone that maintains early pregnancy, which is human chorionic. Excuse me, I won't even try to say it, but it's it's human chorionic gonadotropin hormone, and that's the hormone that excuse me, birth, your pregnancy test. Dang it, your pregnancy test will be detecting. That's what they're looking for in the pregnancy test to determine whether you have a valid pregnancy or not. And again, by the end of this stage, um, implantation has begun, and the foundation for all further development is in place. Excuse me for that little mental hiccup there. I couldn't remember the the name of a pregnancy test. I was going to call it a birth test. Okay, this is figure 23.4, the oocyte with sperm, two cell stage, and the morula. So let's take a look at these three images. And again, they show development in real time. So on the left, you're seeing the oocyte surrounded by sperm just after fertilization has occurred. And this is the starting point. One cell containing a complete genetic blueprint. And then the center image, the zygote divided into the two cell stage. So these cells are called the blastomeres. And the overall size, again, hasn't really changed, just the number of cells. And on the far right is the morula, a solid ball cells formed after repeated division. So by this stage, the developing structure is moving toward the uterus, preparing for implantation. And what matters here isn't memorizing these images, it's recognizing the larger pattern that you see. One cell becomes many without growing larger, so it's just dividing. And the pattern sets the stage for everything that comes next. I know this example I'm going to give isn't exactly related to this, but if you have a pie and you cut the pie into a whole bunch of pieces, you didn't make more pie, you just divided it into a bunch of pieces. Not the best example, but you get the idea. Okay, so we're looking at figure 23.5, stages of early development. So early development, and we're looking at a timeline here. And the figure, it lays out early development as a day-by-day journey. So on day 0, fertilization occurs in the uterine tube. The sperm and the oocyte nuclei, both, remember they're called the pronuclei at this stage, they form, and then they unite and they create the zygote. About 30 hours later, the first cleavage division occurs, and the zygote splits into two cells. And then this is the two cell stage that that's shown there on day 1. I was going to refer back to the last slide as well. And then by day 2, the embryo reaches the four cell stage, and by day 3, continued divisions produce an early morula. Um, and then on day 4, the morula becomes more compact as it moves closer and closer to the uterus, because you got to remember it's still traveling to its destination. So around days 6 to 7, the structure becomes the blastocyst and begins implantation at this point into the uterine lining or the endometrium. And then throughout this entire process, the embryo just is dividing. So it's not growing larger. It's just dividing while it's traveling from the uterine tube into the uterus. And this little timeline helps explain how quickly development unfolds in the earliest days of pregnancy and how precisely it's coordinated. Okay, here we're going to talk about implantation. So, implantation is the moment the pregnancy becomes physically established. It begins when the blastocyst attaches to the uterine lining. Remember, that's called the endometrium. And to do this, the blastocyst releases proteolytic enzymes, and they gently digest part of the urine surface. The outer layer of the blastocyst, which is the trophoblast, that develops these little tiny projections called microvilli, and those microvilli push into the endometrium and they anchor the blastocyst in place. So as implantation continues, the endometrium grows around that blastocyst and it fully embeds itself within the uterine wall. And this whole process typically occurs between the end of the first week and the end of the second week after fertilization. So during this time, the trophoblast releases that human chorionic gonadotropin hormone, or abbreviated small h, capital C, capital G, HCG. And this hormone keeps the corpus luteum functioning and helps prevent the maternal immune system from rejecting the developing blastocyst. So implantation continues. It progresses as the placenta starts forming. Um, it develops from both the endometrium and cells that surround the embryo. It attaches to the embryo, the embryo to the uterine wall, and allows for nutrients, gas, and waste exchange between the mother and the developing embryo. So implantation isn't like just an attachment. It's not just stuck on the wall. It's the beginning of the biological cooperation between the two systems or two organisms. All right, so here we have figure 23.6, implantation. So this is a nice visualization of the implantation. So what we see in the figure is, again, the implantation as a progressive process, not one single event. So in the first stage, the blastocyst is positioned against the endometrium. The outer layer, the trophoblast, is in direct contact with the uterine lining, while the inner cell mass remains protected inside. And the inner cell mass eventually will become the embryo. In the next stage, the trophoblast begins to invade the endometrium. Shouldn't say invade, but it starts infiltrating the endometrium, and the cells extend outward, anchoring the blastocyst deeper into the uterine wall. And the I was going to say invasion again. The invasion allows the developing embryo to access nutrients and oxygen. I keep thinking of the movie Alien. So the implantation, right? What this whole thing is called implantation, but the progression further into the endometrium allows the developing embryo to access nutrients and oxygen. All right, and I'll leave these little blurbs of mine, these little side notes, because hopefully it'll help you remember this particular slide. It's like, oh, that's a slide he talked about aliens. Um, yeah. So your book says invasion. And so in the final image, it shows the blastocyst embedded within the endometrium. And so at this point, implantation is pretty much underway. The trophoblast continues to expand and it differentiates and it starts forming structures that later contribute to the placenta. So when you look at this figure, focus on two things: where the trophoblast is moving and how the endometrium responds. So implantation works because both tissues actively participate in this process. We're looking at figure 23.7, blastocyst implanting in endometrium. So we're looking at the implantation in the tissue, the view of implantation in the tissue. So that's what we're looking at, the tissue level here, implantation. At the top of the image is what's called the uterine lumen, and it's the open space inside the uterus. Beneath that is the endometrium, the thick, again, it's a vascular lining, and that's preparing to support pregnancy. And embedded within that endometrium that you see there indicated by the arrows is the blastocyst. And then notice that it's no longer sitting on the surface, that it's moved into the uterine lining, and the surrounding tissue appears dense and active. That's because the trophoblast cells are, again, according to your book, invading the endometrium, um, merging, I would say, with the endometrium. It anchors and develops the embryo there, and so it's establishing early connections with the maternal blood vessels. And what this image captures is, I guess, a good commitment between the two tissues. At this stage, implementation. I keep wanting to say that implantation is no longer a possibility. It's actually underway, right? So the embryo is physically integrated into the uterine wall, and the maternal tissue is actively responding to support the development of that blastocyst. So this is a microscopic moment when pregnancy becomes firmly established. Again, from Science to Technology 23.2. So let's talk about pre-implantation genetic diagnosis. So pre-implantation genetic diagnosis, or PGD. Right, this takes place after in vitro fertilization and just before implantation. And so at this early stage, a cleavage embryo consists of only a few cells, and doctors can actually remove um, one or two cells and analyze them for genetic disorders. The remaining cells, they just continue to develop normally. Embryos found to be free of any specific genetic conditions can then be implanted into the uterus. So PGD is something that's commonly used to identify serious inherited illnesses such as cystic fibrosis or certain forms of anemia. And it allows families to reduce the risk of passing on devastating diseases. In some cases, PGD is also used to identify an embryo that may later provide um, umbilical cord stem cells. And so after birth, those cells can potentially be used to treat a sibling or something like that. I think there's a movie about that, like My Sister's Caretaker, My Sister's something. Anyway, oh, we'll talk about that in class. Then, yeah. So instead of looking for like a match recipient, they can actually identify a sibling very early on at this stage. Um, and in the movie, just not to get off sidetrack, but in the movie, an a younger sister was conceived specifically for that purpose of being a transplant for her older sister. So it's a pretty good movie. Anyway, get back on track. So PGD again highlights modern reproductive technology, and again, this is beyond conception. Is we're talking about the intersection of genetics, medicine, and especially ethics. Once you get into the stage, and the movie does a real good job of highlighting the ethical dilemmas with having access to this type of technology. And again, all this is done before pregnancy even begins. Okay, this is figure 23B, pre-implantation genetic diagnosis probes disease causing genes in an 8-celled cleavage embryo. So what we're seeing here is a visualization of PGD. The figure shows how pre-implantation genetic diagnosis, again, or PGD, ends up working at the cellular level. So at the top, you're going to see an 8-cell cleavage stage embryo. And at this point, remember, one cell can safely be removed for genetic testing without disrupting normal development. And that single cell ends up being analyzed using DNA probes, and they look for specific genes associated with inherited disease. So if the embryo is found to be genetically healthy, it's allowed to continue developing and can be implanted into the uterus. The remaining cells, again, are capable of completing normal development. So if a disease causing gene is detected, though, the embryo, again, is not implanted. And so what the figure ends up emphasizing is mainly the timing. The PGD happens before implantation, hence pre implantation. Again, long before pregnancy is established, and this allows genetic information to guide reproductive decisions early. Okay, let's go ahead and talk about extraembryonic membrane formation and placentation. So, extraembryonic membranes and placentation. So, once implantation begins, the body starts building the structures that are going to support the embryo. The chorion, which is the outermost extraembryonic membrane, right? It forms from the trophoblast and an additional layer of cells that line it up. You have these little finger-like projections that go outward from the chorion and they invade the endometrium, and these end up becoming the chorionic villi. Then we have spaces called lacunae, and they form in the endometrium around the chorionic villi, and the spaces fill with maternal blood. And this allows exchange to begin without the mother's and the embryo's blood directly mixing. So, as development continues, this embryonic blood vessels they start growing within the chorionic villi, and these vessels connect the embryo through a stalk that later ends up becoming the placenta. And inside that villi, or inside the villi, you have a little network of capillaries, and they are what's used to support nutrient exchange, gas exchange, waste exchange. The amnion that forms next, and this is the second extra embryonic membrane. So that develops around the embryo during the second week. And the amnion encloses you guessed it, the amniotic fluid. And the amniotic fluid, this cushions and protects the embryo. It absorbs shock. It helps maintain basically a stable temperature. And together, these membranes, they don't become the embryo, but they make the embryo life possible. It's the penthouse suite for the embryo. Okay, so we are going to continue talking about the extra embryonic membrane formation and placentation. This is part two of that. Again, so as development continues, the amniotic cavity starts expanding. It grows large that the amnion eventually contacts the chorion, and the two membranes fuse to form the amniochorionic membrane. And so another structure is the yolk sac, right? The extra embryonic membrane forms during the second week beneath the embryonic disc, and the yolk sac plays an important role in the blood cell formation. Week three, a structure called the allantois. This develops between the yolk sac and the connecting stalk. And the allantois, I don't know if that's French, allantois, that it contributes to the formation of the blood cells and the umbilical blood vessels. And those will eventually connect to the embryo to the placenta. The placenta itself forms where the chorion contacts the uterine wall, and it contains both the embryonic and the maternal components. So this creates a shared interface without mixing blood. And at the interface of the placental membrane, you're going to have a thin barrier that separates the embryonic blood from the maternal blood, and again, allowing oxygen, nutrient, and waste to move between them. And that's made from it's a lot of detail, but it's made from the endothelium of the embryonic capillaries and the epithelial wall, the chorionic villi. And these structures, they create that lifeline that supports growth throughout pregnancy. Now, I say there is a lot of detail here, but I'm saying all this to kind of give you an idea of how important it is, what creates that barrier between the maternal and the embryo um, blood connection to separate the blood. We say those things, but you don't realize all the detail that's involved uh, to minimize what crosses over the placenta. Okay, we're looking at figure 23.8, extra embryonic membrane formation. Again, we're looking at a visualization of the extra embryonic membranes, the figure. So what this figure does is it brings everything together. It brings all the extra embryonic structures in one particular view, one single view. And let's go ahead and start from the outside. So we have the endometrium, which lines the uterus, and just inside is the chorion, the outermost embryonic membrane. Extending from that chorion are the chorionic villi. Remember, those are used to help anchor the developing structures, and they help support the exchange with the maternal blood. Inside the chorion is that extra embryonic cavity surrounding the developing embryo and the membranes. And let's go and move a little bit further inward or center. The embryo itself is formed from the embryonic disc, and that's made up of three primary germ layers. Now, you may have heard of these germ layers like in a very basic reproductive course, but this is how we get there at the cellular level. And the three layers are the ectoderm, the mesoderm, and the endoderm. Hopefully, these might sound familiar from something a long time ago that you may have studied. And the layers give rise to all tissues and organs. So, above the embryo is the amnion, and that encloses the amniotic cavity that's filled with that protective fluid. Below the embryo is the yolk sac, which plays an early role in blood cell formation. Connecting that embryo to the chorion, remember we said, is the connecting stalk, and that ends up later becoming the umbilical cord. And just looking at this figure, just think layers and spaces, right? Layers and spaces. So the embryo sits at the center, it's supported on all sides by the membranes that protect, nourish, and connect it to the maternal environment. Figure 23.9, development of the amnion and umbilical cord. Okay, so what do we look in here? What are we visualizing here? So this figure shows the embryo and how it becomes fully enclosed and connected to the placenta. So as the amnion expands, it grows around the embryo, creating that amniotic cavity. And remember that amniotic cavity is filled with that protective fluid. And then over time, the amnion surrounds not just the embryo, but also the connecting stalk. The connecting stalk contains those blood vessels that were developed from the allantois or the allantois. It's Allantois, if you can't see that on the slide. Pronounce it how you will. And then the amnion wraps around this. So the stalk ends up becoming the umbilical cord, which is, you know this one, the embryo's lifeline to the placenta. And on the placental side, the chorion forms branching chorionic villi. Remember, those extend into the endometrium. And these villi are surrounded by maternal blood, and that allows exchange um, to occur without having any direct blood mixing. So next is the placenta, right? Continuing on with the placenta, that continues to develop at this interface. It links the embryonic blood vessels in the villi to maternal circulation in the uterine wall. And so, one thing to take away from this figure, what it's emphasizing, is the enclosure and connection. It's a good way of just looking at this figure. The amnion protects, the umbilical cord connects. I'm not trying to rhyme, it just happened. And the placenta sustains. So, again, these structures make continued growth possible. But they would make a good wrap. Amnion protects, the umbilical cord connects. Okay, we're looking at figure 23.10. The placenta consists of an embryonic and a maternal portion. So the placenta, right? So we're looking at two systems here. Two systems, one organ. And what we see here in the figure, what it's highlighting, is the idea that the placenta is made up of two parts, one embryonic and one maternal. At the top, you see the umbilical cord connects the embryo to the placenta. Inside the cord, you see two umbilical arteries carrying, waste-filled blood away from the embryo, and one umbilical vein carrying oxygen, nutrient-rich blood back. So the embryonic portion of the placenta comes from the chorion. It forms the branching chorionic villi, and these contain the embryonic blood vessels. I've said this like 4 or 5 times already. So it must be very important. It might be like on a quiz or a test or something, or it might be a question I select. Yeah. And so what surrounds these villi are the spaces that we call the lacunae, and they're filled with the maternal blood. So lacunae filled with maternal blood, and the blood comes very close, right? Maternal and embryonic blood come very close, but they don't mix. The maternal portion of the placenta is the decidua basalis. It's a specialized region of the endometrium. And maternal blood vessels open into that lacunae that we talked about. They supply oxygen, they supply nutrients, but the two circulations that are in the placental membrane, you have a thin layer that allows exchange while keeping the two blood supplies separate. And what we're seeing here in the figure, what we're highlighting in the figure, like I said before, we're looking at the placenta for kind of what it really is, the carefully designed interface that allows two independent circulatory systems to work together between two different organisms. Right. Again, I know most of you have heard this before, how the mother circulatory system doesn't make contact with the fetus, and it doesn't, or the embryo in this case, and it protects the baby. Um, Now you kind of see why, like you see some of the technicalities of it. Um, Sit back after this slide. Don't go to the very next one. Just absorb that amazing miracle. Okay, we're looking at figure 23.11, the placental membrane, the endothelium of an embryonic capillary, and the epithelial wall of a chorionic villus. So I'm just reading the caption. So let's continue. So placental membrane, which is the exchange barrier. And what we see here in the figure is the placental membrane, the thin boundary where the exchange actually occurs. So inside those chorionic villi are embryonic capillaries. Remember, they carry the embryo's blood, and surrounding the villa are the lacunae filled with the maternal blood. Um, and so the two blood supplies, they come extremely close. They never mix, and they're separated by that placental membrane. And the membrane is made up of two layers. We'll get into those. The first one is the endothelium of an embryonic capillary and the epithelial wall of the chorionic villus. So across, they're listed up there on the slide, and across this very, very thin barrier, oxygen and nutrients can move from the maternal blood into the embryonic blood, while carbon dioxide and the waste move in the opposite direction. And so the design is very precise, and it's close enough for efficient exchange, but you have it's structured enough yet to keep the two circulatory systems separate. So without completely mixing, it's close enough so there is a small barrier there, yet efficient enough to provide exchange of the right amount of waste being removed and the right amount of oxygen to be transferred into the embryonic side. And so the membrane is, again, that final checkpoint between maternal and embryonic circulation. And again, this is very, 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 very essential for survival throughout pregnancy. Okay, let's go ahead and talk about the embryonic stage. So the embryonic stage runs from the third week of prenatal development through the end of the eighth week. And again, this is the most critical period of development because this is when the basic body plan and all the major organs form. So you have two major processes, and we define those by stages: the gastrulation and the organogenesis. So gastrulation is that movement of cells within the embryonic disc that creates multiple layers. And by the end of the second week, the disc has two layers: the outer ectoderm and the inner endoderm. But during gastrulation, you get that third middle layer that forms between them, and that's called the mesoderm. You should kind of know these already from medical terminology. So these three layers are called the primary germ layers: the ectoderm, mesoderm, and the endoderm. And every organ and tissue in the body develops from one of these layers through a process called organogenesis. So an embryo that has completed gastrulation is called a gastrula. And from this point forward, the basic structure of the human body begins to actually take shape and take form. Okay, here we have figure 23.12. Organs form from primary germ layers. So we said the germ layers of the body's blueprint, right? So we see the entire human body form from just three primary germ layers: the ectoderm being the outer layer, mesoderm middle layer, the endoderm the inner layer. The ectoderm gives rise to the nervous system. This includes the brain, spinal cord, also the skin, sensory organs, hair, nails, and skin glands. The middle, I was going to say middle layer, the mesoderm, this gives form to muscle, bone, and connective tissue, and blood vessels, lymphatic vessels, kidneys, reproductive organs, and such. The endoderm, that very inner layer, it ends up becoming the epithelial lining of the digestive and the respiratory tracts, and other organs like the liver, pancreas, gallbladder, and the urinary bladder. So everything you see in the adult body can be traced back to one of these three layers. And when you look at this figure, don't think memorization. So don't try to memorize just the picture. Think about the origin of where these things came from, and the structure will follow. Will follow. Table 23.1: Stages and Events of Early Human Prenatal Development. Okay, let's talk about gastrulation and organogenesis in a little bit more detail. So, why gastrulation even matters? Gastrulation is one of the most important events in human development because it determines cell fate. Right? So once the cells move into a specific germ layer, they're set, their future is set. They're going to be something. So when cells in the ectoderm and the endoderm, they become epithelial cells, while cells in the mesoderm, or they become connective tissue. The remarkable thing is that the primary germ layers, they retain stem cells. So this is why, like even in adulthood, tissues can grow, repair, regenerate, and each germ layer gives rise to specific structures. And repeated, I'll repeat this again. Ectoderm, we said, forms the nervous system, parts of the special sensory organs, the epidermis, which were things like, include the things that are like hair, nails, skin glands, the linings of the mouth, and the anal canal. The mesoderm, the middle layer, the mesoderm cells, we said they form the muscle, bone, blood, the blood vessels, the lymphatic vessels, um, the kidneys, the internal reproductive organs, and also the epithelial linings of the body cavities. And then we have the endoderm cells, and they form organs, like we said before, the pancreas, gallbladder, digestive tract, respiratory tract, urinary bladder, urethra. We also have something called um, something that the embryo can be um, vulnerable to, something called teratogens mentioned on this slide. So I was trying to segue into that very last bullet. And teratogens are, again, like it says there, they're factors like drugs, chemicals, infections, or radiation. And what they do is they'll cause malformations uh, by disrupting development during periods of like rapid growth and specialization of those cells. Um, so teratogens again are things that can you don't want. But overall, just to kind of like surmise this whole slide, gastrulation doesn't just shape the embryo; it kind of sets the foundation for the entire body. There you go. My closing thought. Okay, let's look at figure 23.13, the embryo at 3.5, and 4 weeks. Um, so we're looking at rapid change from weeks 3 to 4 here. And what you see here on the image is just how quickly development accelerates during the embryonic stage. And so you see the picture on the far left, the embryo is at 3 weeks. And at this point, the body is still relatively flat. The nervous system's beginning to form. The early segmentation is visible, but the overall structure is very simple. The center image you'll see at 3.5 weeks, the embryo begins to fold. And the folding is critical because it transforms that flat disc into this three-dimensional body, and you can start to see the head region and the enlarging of the body as it curves. By week 4, which is the image on the right, the embryo looks way different. Uh, the body of the embryo begins to have this C shape. The head is very prominent. You can see the early limb buds popping out, and you'll see internal organ regions and they'll become visible. And what's pretty striking here is not the size, but the organization of the structure, the formation. So in just this 1 week, from week 3 to 4, the embryo shifts from a simple form to a recognizable human body. And this is why the embryonic stage is so sensitive and so important. Okay, we're looking at figure 23.14, embryonic development. Now we're looking at weeks 5, 6, 7, and 8. So embryonic development 5 through 8. So between these weeks, 5 and 8, development shifts into like high gear. Early in this particular period, the embryo, again, it starts off with that curved C-shaped body. The head grows rapidly as the brain starts developing. Well, not starts, but continues to develop. You'll see the eyes, ears, and you'll start seeing facial structures begin to take like a recognizable form. The limb buds that we mentioned earlier, they start getting longer. They flatten into paddle shapes. And as the weeks pass, the paddles divide into the digital rays. And by the end of this stage, you'll see little fingers and toes separation, the separation of fingers and toes. Now, the internal organs are forming and they're repositioning. That includes the heart, which is it's very prominent early on and continues to refine its structure. And by the end of the 8th week, Nearly all of the major organ systems are present in this basic form. And so, even though the embryo is still small, the human body is complete. And so, what this marks is the transition to the next phase of development, which is growth and maturation. Right? Okay, let's look at figure 23.15 and 23.16, embryonic development and critical periods in a development. So again, critical periods in development. What we see here in these images, or what they illustrate, is an essential concept in embryology, which is the timing, right? So timing matters. Um, on the right, you see uh, the top graph on the right. This shows when like major physical structures develop and different organ systems form during very specific windows of time. So we know this to, a day. The heart, limbs, eyes, ears, they all develop very early within the first few months. The reproductive systems, they develop later. Uh, and the central nervous system, that develops early and continues maturing throughout pregnancy. Um, the windows are called critical periods. And these are, again, times when the doctor is going to be checking and looking at a normal development of these structures. And also looking at anything that, if there's any disruption to the development of these structures. So those of you who've had children, have gone to the doctors, and you've gone through these particular periods where they're looking at development at a certain stage, I mean, we know what should be there for a viable pregnancy. Would you see the log graph there? It shows different when different teratogens cause the most damage. So again, certain drugs or chemicals can interfere with development only if exposure occurs during that specific window. So, for example, thalidomide causes limb defects if exposures happen early. And there's a documentary on this. I don't remember it, but I'll mention it during class. Then we have diethylstilbestrol, or DES. Again, this affects reproductive development later in pregnancy. Accutane interferes with multiple systems if the exposure occurs during the early organ formation. And so, what we see there on the image on the left, that reinforces the idea just but visually. So you have an embryo that's developing inside the amniotic environment. It's protected, but again, it's still vulnerable. So I said before, there's a there's a barrier to protect the embryo or the fetus, but not against everything. But again, the takeaway from this slide is that the same exposure can have very different effects depending on just when it occurs. So timing is very, very important. And so that's why understanding developmental timing is just as important as understanding the anatomy. Let's talk about our clinical application 23.1. I could have sworn I already said 23.1. But no, it's probably not. I'm probably thinking of section 23.1. So this is our first clinical application then, 23.1. Clinical application. And this slide talks about the causes of birth defects. So the this again, it highlights how environmental factors and exposures can disrupt normal development, especially during critical periods. Again, one of the most well-known examples is thalidomide, and it was once prescribed as a mild tranquilizer, and it was later found to cause severe limb malformations if taken early in pregnancy or uh, during that critical window for limb formation. Again, there's an entire documentary just on this. Rubella, or again, German measles, that's another powerful teratogen. Again, the type and severity of birth defects depends on when exposure occurs. But again, vaccination has dramatically reduced the impact of this. Alcohol exposure, I think we all know this one. Alcohol exposure during pregnancy can lead to what's called fetal alcohol effects or fetal alcohol syndrome. And this includes abnormal facial features, slowed growth, and impaired intellectual development. Cigarette smoking. Some of these don't have to be said, but I mean, it's for the class. So, Cigarette smoking reduces the amount of oxygen that gets delivered to the fetus, and so this can result in poor growth, low birth weight, stillbirth, or spontaneous abortion. And then continuously on a day-to-day uh, um, on a day-to-day basis, that's what I was looking for. Nutrition. Again, we covered nutrition a couple of chapters ago, but nutrition matters here. So malnutrition can slow fetal growth and permanently alter metabolism, which increases the risk of things like obesity or type 2 diabetes later in life. And exercise. So excessive intake of certain nutrients. If people are exercising during pregnancy and they're taking too many nutrients, vitamins, and stuff, that could actually be harmful. So imagine like a bell curve in the middle where you have the right amount of nutrients and not too much, bad stuff, malnutrition, but also trying to overdo it with all the what people think is the good stuff. You can also have occupational hazards, and this can include things like chemicals in the workplace. Every workplace that you go to with any chemicals, they should have MSDS sheets, and you should be, if you are pregnant and you're working anywhere, and you might just want to ask for that. It's called the Right to Know Act, and so there should be an MSDS location of every chemical there, and just go through and make sure that there's nothing that would provide or present itself as a teratogen to cause a birth defect. But again, these occupational hazards can lead to anything from just a birth, not I should say just a birth defect, but a birth defect or to pregnancy loss. We have other things like the Zika virus, as mentioned in your book. And again, this is something that's transmitted by mosquitoes. It's also carried in semen, and it can cause severe brain abnormalities if exposure occurs during the first or the early second trimester. Because remember, the nervous system is developing. So this leads to or can lead to microcephaly. And so the common thread across all of the examples we mentioned on this slide, um, that exposure matters, but when the exposure happens also matters. Hmm? All right, here we're going to talk about the fetal stage. Right? So the fetal stage begins at the end of the 8th week of prenatal development, and that continues until birth. So in this stage, it's defined less by new structures and more by the rapid growth and refinement of the structures. So we're looking at body proportions, how they change. They change dramatically as organs start maturing and the system starts preparing for life outside the uterus. Sounds like a TV show. Life outside the uterus. I'm sorry for my bad jokes. I've watched too much reality television. So let's go back to the lecture. Again, starting off at the fetal stage. Let's go back to the fetal stage. The head is relatively large, and the lower limbs are short. We mentioned before they were paddling at the last stage, but during like the 3rd month, the body starts lengthening quickly. The head grows slower in proportion, and the upper limbs um, they they reach the proportions that they're going to keep throughout development. Right? Not their total length, but like proportion to the body. And once it's in that proportion, then it's development. But by what's this here, week 12, the external reproductive organs can be distinguished as male or female. So this is when you get the ultrasound and they can tell you, yep, it's a male or it's a female. During the 4th month, the lower limbs lengthen, the cytoskeleton continues to ossify, the ossification, if you remember that from your musculoskeletal chapter. And what happens is cartilage starts being replaced with bone. By the 5th month, you have the overall growth. It starts slowing down. Hair begins to grow, and the fetus starts making muscle movements. So you'll start seeing the few muscles and twitches and stuff like that. Those of you again who've had pregnancies, and this stuff is nothing new. It's pretty cool to me though. I've actually seen somebody. I got to see the little baby in real life, not like in a movie. The little baby kick through the skin. You see the little. I'm assuming it's a foot. Um, you see you see the protrusion, like in the movie Aliens. All right, I better stop with the jokes and continue with the lecture. Where are we at? We ended up with month 5 or month 6. So the fetus starts gaining weight. Um, the skin is kind of thin and wrinkly, like an old person, right? Um, but not old, right? Just new skin. By the 7th month, fat is actually starting to deposit, and the skin becomes a little bit smoother, right? And the eyes actually start opening up. And then the final trimester, this is where the organs continue to specialize, they continue to grow. Brain cells form increasingly complex networks, and the digestive and respiratory systems are the last to fully mature. And by the end of this stage, focus is again no longer on forming of the body, but preparing it to function independently, like we said in our reality show, Life Outside the Uterus. Now playing Mondays and Fridays on ABC, something like that. No, it'll be an HBO special. This is figure 23.17. Body proportions change over a lifetime. Well, look at that little alien embryo looking baby. Alien as in outer space alien. Um, yeah, body proportions across the lifespan. So what we see here in this figure is how the body changes from early development through adulthood. So early on, again, all the way back to the embryonic and fetal stages, the head makes up a large portion of the total body length, and you can't deny that does look like the typical alien from a alien TV show. But anyway, but back to human development. What we see here is like a rapid development of the brain, the nervous system. At birth, the head again is still proportionally large, while the trunk and the limbs are relatively short. And as the childhood grows up, progresses, the trunk actually lengthens, the arms and legs grow faster, and the head grows, but at a slower rate as far as volume is concerned. What's happening in the head is a lot more neural connections are happening within the brain. So it's not like nothing is happening in the head. There's a lot growing. There's a lot of development within the head, which is all in the cranium, even though the head itself isn't getting bigger. So yes, arms and legs are physically growing longer and faster than what the head appears to be, but as far as development, the brain is developing significantly fast. Oh, and let's continue to adolescence, right? So the growth again accelerates again. The limbs uh, grow pretty fast. This leads to the final like adult proportions. And when you get to adulthood, the head represents by volume, by proportion, it represents a much smaller fraction of the total height. But the the legs, and while the legs make up a larger share, I'm still trying to like make sure I emphasize that yes, the head represents a smaller fraction of the total height of the height of the organism, in this case, a human. A lot of development has actually been taking place. So, not to confuse development with growth in this case. Yeah. Um, and so, again, the key idea here is differential growth. Not all body parts grow at the same rate or at the same time. And what we see here in this pattern is looking at how to explain why infants look different from children and why children look different from adults despite having the same body structures. Okay, here we're looking at figure 23.18, formation of external reproductive organs. So formation of external reproductive organs, um, yeah, so we see the external reproductive organs develop from the same early structures, and then they diverge into either male or female forms. So again, in early development, all the embryos have identical external structures, what's called the genital tubercle and the urogenital folds, and the labial scrotal folds. So just in case these are on the test, genital tubercle, the urogenital folds, and the labial scrotal folds. They're identical. But at this stage, the external sex differences, again, they're not very visible. They're not different. But as development progresses, as the formation moves, changes, I feel like I'm about to do a Jeff Goldblum impersonation any moment now. But these structures, they respond to genetic signals and hormones. So um, within the males, the genital tubercle elongates to form the penis, and the urogenital folds fuse around the urethra. The labial scrotal folds fuse to form the scrotum. Right? But within females, that genital tubercle develops into the clitoris, and the urogenital folds remain separate and became the labia minora, while the labial scrotal folds become the labia majora. So this shared origin explains the similarities and differences that we see in the adult anatomy. Figure 23.19, figure 23.19, also why I say 19. The full term fetus positioned with head near the cervix. And by this point, you've already done this lab in the lab section of the course. So the full term position. So what we see here is a full term fetus in its position for birth, right? So notice that the fetus is curled in a head down position with the head located near the cervix. And this is the most common and what we believe to be the safest position for delivery. And we call this the cephalic presentation. I'm pretty sure that term was on your lab. Surrounding the fetus is the amniotic fluid. Again, that was used to cushion the body and it allows limited movement. The umbilical cord we still see here connects the fetus to the placenta, and that remains attached to the uterine wall and continues with the nutrient, gas, and waste exchange until delivery, until birth. The uterine wall we see here encloses and supports the fetus still, while the cervix forms the lower opening of the uterus. So as labor begins, the cervix begins to soften. It starts thinning out. It starts dilating. This dilating is what you're more familiar with, and this is going to allow the fetus, excuse me, to move into the birth canal. And so what we see here on the image is the final stage of prenatal development when growth is complete and the body is positioned for the transition to, again, our HBO special, life outside the uterus. Table 23.2, stages of prenatal development. Okay, let's go ahead and talk about fetal blood and circulation. So, during fetal development, the fetus does not use its own lungs for oxygen exchange. Instead, maternal blood supplies oxygen and nutrients. Not only that, it removes waste through the placenta. So, exchange happens across the placental membrane and the umbilical vessels, which carry blood between the placenta and the fetus. So fetal blood is especially well adapted for this job. So fetal, excuse me, I have to really enunciate these. Fetal hemoglobin, um, excuse my accent when I don't pronounce the words correctly. Fetal hemoglobin, that carries 20 to 30% more oxygen than adult hemoglobin and allows efficient oxygen uptake even at lower oxygen levels. So, oxygen rich blood travels from the placenta to the fetus through the umbilical vein. And so, most of this blood bypasses the liver through a special vessel called the ductus venosus. And we can point that out in the fetal heart. And so, the ductus venosus connects the umbilical vein directly to the inferior vena cava, allowing oxygen rich blood from the placenta to mix with the oxygen poor blood returning from the lower body. And this mixed blood then returns, I'm sorry, not returns, that enters the right atrium of the heart and it continues throughout the unique pathways of fetal circulation. And so here the key idea is the efficiency of this, where fetal circulation is designed to deliver the most oxygen possible to critical tissues, again, especially the brain, before birth. All right, let's go ahead and continue talking about the fetal blood and circulation, and let's focus on the bypass system. So, this is really, really cool, by the way, this system. So, fetal circulation is built around bypasses because the lungs aren't developed yet. So, the first bypass is the foramen ovale, and that's an opening between the right and left atrium. And most blood entering the right atrium, it's shunted directly into the left atrium, and this allows oxygen-rich blood to bypass the lungs. If you can remember, in normal blood circulation for an adult, right, the blood uh, uh, moving from, the lungs into the right atrium, into the ventricle, back out to the body. So what we're doing here is just basically bypassing the lungs. Right? So you get a small amount of blood. It does enter the pulmonary trunk, but only enough to kind of nourish and help the tissue develop. So more for its development. And then the secondary bypass, right, is the ductus arteriosus. And I know it sounds like a Harry Potter word. Ductus arteriosus. All of these sound like Harry Potter words. Uh, and so this is the vessel that connects the pulmonary trunk directly to the aorta. And what it does is it diverts most of the blood away from the lungs. And so as a result, blood in the aorta for the fetus contains a mixture of oxygen rich and oxygen poor blood. And some of this blood supplies the lower parts of the fetus, while the rest enters the umbilical arteries. And the umbilical arteries, remember, carry blood back to the placenta where carbon dioxide and waste are removed and oxygen is replenished. So these two shunts, the foramen ovale and the ductus arteriosus, create an efficient circulatory system that's tailored for life right before birth. So life not outside the uterus. And a really cool thing happens at the instant of delivery when the baby takes its first breath. But I don't want to ruin it and give you any spoilers. And so I will wait until that slide. Transitioning from to life outside the uterus. HBO special, now playing. Figure 23.20. Fetal blood in circulation. Oxygen and nutrients diffuse into the fetal blood from maternal blood, and waste diffuse into maternal blood from fetal blood. All right, so this is the exchange at the placenta, which sounds like a cool place to hang out. Exchange at the placenta. So what we see here in this zoomed in picture is where exchange actually happens between the mother and the fetus. Right, so the maternal blood flows through spaces and the placenta. Remember, we called those the lacunae. That word sounds Hawaiian to me. I'm going to be making a lot of not so funny jokes, but just a little things that help me remember these things. So the lacunae, and what surrounds, they surround the chorionic villi. So inside each of those villi are those fetal capillaries we mentioned that carry the fetus blood, and across the thin placental membrane, you have oxygen and nutrients that diffuse from the maternal blood to the fetal blood. And at the same time, you have again, carbon dioxide and metabolic waste that diffuse from the fetal blood into the maternal blood. And so it's the mother who gets rid of the waste for the baby. But notice that the blood supplies again remain separate. They're not impenetrable, but they do remain separate. They are very, very good, but um, not 100%, right? But as close as you can to that. And so what happens, exchange happens by diffusion here, not by direct mixing. And so blood that has delivered oxygen to the fetus, that returns to the placenta through the umbilical arteries. And after reoxygenation and nutrient uptake, blood travels back to the fetus through the umbilical vein. And this figure just kind of reinforces that central idea of fetal circulation. The placenta functions as the fetus's lungs, kidneys, and digestive system all in one. Okay, we're looking at figure 23.21. 23.21 pattern of fetal circulation. So again, the figure here puts the whole fetal circulation pathway together. We start off with the placenta where the blood is oxygenated, and the oxygen-rich blood travels to the fetus through the umbilical vein. Most of it bypasses the liver through the ductus venosus, one of the bypasses, remember that, and enters the inferior vena cava or inferior vena cava, and that heads straight to the heart. So when blood reaches the right atrium, it follows again another unique fetal shortcut. Instead of going to the lungs, most of it passes through the foramen ovale, moving directly into the left atrium, and from there it enters the left ventricle and it's pumped into the aorta, and that supplies the brain and the upper body with the most oxygen-rich blood available. Remember, the blood that does not enter the right ventricle flows into the pulmonary trunk, but because the lungs are not functional, most of the blood that's diverted through there goes through what's called the ductus arteriosus and directly into the aorta. So from the aorta, blood supplies the lower body, then returns to the placenta through the umbilical arteries, where there again it is reoxygenated. And one big takeaway for this figure again is efficiency. Did I say it correctly? Is efficiency. Sometimes I kind of blend my words together. And so fetal circulation uses three shunts. We said the ductus venosus, the foramen ovale, and the ductus arteriosus. Um, yeah, and we use this to one bypass the liver and lungs, and to prioritize oxygen delivery to the vital organs. And the entire pattern, spoilers, will change dramatically at birth, but I won't, I won't spoil it. Okay, here we're looking at figure 23.22, schematic diagram of fetal circulation. So we're looking at a flow chart basically and trying to get the big picture just through a flow chart. And what we see here is like a schematic uh, strip of the fetal circulation broken down into its essential pathways. You see the oxygenated, nutrient-rich blood that leaves the placenta. It enters the fetus through the umbilical vein. We said some blood passes through the liver, but most bypasses it via the ductus venosus, and then it flows into the inferior vena cava or vena cava, however you want to say it. And from there, the blood enters the right atrium instead of following the adult pattern. Most of it passes through the foramen ovale into the left atrium, then into the left ventricle, and out through the aortic arch. So delivering, again, the most efficient rich blood to the heart, brain, and the upper limbs. But the blood that enters the right ventricle flows to the pulmonary trunk. And because, again, the lungs are not functional, I'm kind of repeating myself. I'm saying this one way with the picture, one way with the flow diagram, but I'm repeating the same information. So if it sounds to you like you've heard this already, um, the book gives you different diagrams and images um, to kind of try to remember it. But we said, again, the lungs are not functional in the fetus, and most of the blood is diverted through the ductus arteriosus into the aorta. The aorta then again supplies um, the trunk and lower limbs before returning to the placenta before the umbilical arteries carry carbon dioxide and waste. What this diagram emphasizes are those three key fetal shunts: the ductus venosus, the foramen ovale, and the ductus arteriosus. And together they create a circulation pattern designed for life before birth, life inside the uterus. So efficient, it's protective, and it's again temporary until the baby's born. This is table 23.3, fetal cardiovascular adaptations. Okay, let's go ahead and talk about maternal changes during pregnancy. So, kind of summarizing the slide, what's happening? Maternal changes during pregnancy. We're talking about, in this slide, hormonal control. So, pregnancy, again, there's a lot of hormonal changes that support the developing fetus and the mother's body for pregnancy. So, early in the pregnancy, the embryo secretes human chorionic gonadotropin, or HCG. Again, this hormone is actually important to maintain the corpus luteum. So it's not just being released, it's released for something important. And it continues to release estrogens and progesterone. And what these hormones do is they prevent ovulation by inhibiting FSH and LH, if you remember those follicle stimulating hormone and luteinizing hormone. So they effectively stop the normal reproductive cycle. It's also how some older birth control pills worked by stopping the normal reproductive cycle, um, by tricking the body to thinking it was pregnant. Basically, it's what the earlier um, birth control pills did. So again, during the first 2 months, that HCG levels they increase, they remain high. After that, the HCG starts declining as the placenta takes over production of the estrogens and progesterone. So once that is fully established, the placenta releases large amounts of these hormones, the estrogen and progesterone, and it does that to maintain the uterine lining, and it takes over from there on out. And it also inhibits uterine contractions. It promotes the enlargement of the reproductive organs, and yeah, so very, very important how all this kind of takes place. Another placental hormone that's happening here during maternal changes and pregnancy are placental lactogen, right? So again, that I said there in the slide, it stimulates breast and mammary gland development, and it prepares the body for lactation. The next one is the next hormone is relaxin, and that's produced by both the corpus luteum and the placenta. Okay, I'm just trying to summarize the slide. I'm trying not to read this slide. And what that does is it helps prevent early contractions, and relaxes pelvic ligament. So what this does is it allows for later childbirth. And on top of those hormones, you got a lot of support. You got hormones that support maternal physiology. So you have aldosterone, and that promotes sodium and water retention to increase blood volume, while the release of parathyroid hormone, or PTH, that helps maintain adequate calcium levels in the mother's blood. So all of these hormones or hormonal shifts allow pregnancy to be sustained safely and effectively. Okay, we're looking at figure 23.23, preservation of the uterine lining during early pregnancy. So, what we see here in this figure is a simple 1, 2, 3, 4 step there, but simple but essential hormonal chain reaction. Again, excuse my, I have a slight accent. So anyway, what happens here is all of this begins with the trophoblast cells of the developing embryo, and the cells secrete human chorionic gonadotropin, or HCG. Now, again, just another piece of trivia, like I mentioned before, HCG is what the pregnancy tests, I think, forgot the word again, what they test for to determine whether someone is pregnant, or they just have a high level of HCG. Um, High presence of HCG in males tends to point to a cancer, or like a gonadal tumor. Anyway, the presence of this HCG in females signals the ovary to maintain the corpus luteum, and that prevents it from breaking down as it normally would at the end of a menstrual cycle. So the corpus luteum continues to release estrogens, progesterone, and again, these hormones are critical to promote growth, development, maintenance of the uterine lining. And what this does is it ensures that the environment needed to support pregnancy remains intact for the remainder of the pregnancy. So this hormonal loop prevents menstruation, It stabilizes the uterus during the earliest and most vulnerable periods and phases of pregnancy to sustain a safe and healthy pregnancy. Okay, here we are looking at figure 23.24, maternal hormone concentrations during pregnancy. So, hormone levels across pregnancy. So, what we see here is a graph that shows maternal hormone levels that change over time during the pregnancy. I have to say those words slowly, or they kind of merge. And anyway, I'm trying to enunciate for you very, very clearly. So early on, the HCG, it might be easier for me to say human chorionic gonadotropin versus HCG. Anyway, this rises rapidly, and it peaks during the first 2 months. And this is why HCG, again, I said, is used in pregnancy tests. It's a key indicator that points to you being pregnant. And like I said, again, in males, it's a indicator that they have cancer or a tumor. But again, in females, its main role is to maintain that corpus luteum early in the pregnancy, and then after that peak HCG, the levels decline because the placenta starts taking over hormone production. So again, HCG falls, estrogen and progesterone levels steadily rise through pregnancy, and these hormones are produced primarily by the placenta and are responsible for maintaining, again, the uterine lining, the supporting fetal development, and preventing uterine contractions. And later months in the pregnancy, those estrogen and progesterones end up dominating while HCG remains presently low. Did I say presently? Well, it means it starts dropping, right? Stably. That's what I mean. It's stable and low. And yeah, the takeaway here is that there's a shift in control. It would be a nice key takeaway from this slide. From early ovarian support, which is driven by human chorionic gonadotropin hormone, to long-term placental hormone regulation. Table 23.4, hormonal changes during pregnancy. Okay, let's go ahead and talk about maternal changes during pregnancy, or continue talking about maternal changes during pregnancy. We're going to look at physical adaptations. So, again, as the pregnancy progresses, the uterus enlarges, and what this does is it pushes against the surrounding abdominal organs. And the displacement leads to uh, smaller, more frequent meals. Um, it leads to things like heartburn, increased urinary frequency. Remember, it's pushing against the bladder, so there's less um, volume that can be occupied by the bladder. And so the fetus continues to grow. It demands more. So more oxygen is needed. More waste must be removed. And so in response, the mother's body increases blood volume, increases cardiac output, uh, breathing rate increases, urine production actually increases because now the mother is removing waste um, to keep up with the baby's demands and or the baby's needs, right? And the mother's needs as well. So nutritional demands, again, obviously those increase as well. So food intake increases, um, and it increases to provide, the higher demand for, say, vitamins, minerals, proteins that are needed for normal growth and development of the fetus, as well as maintain the mother's health. So if the diet ends up being inadequate, the nutrition for the mother ends up being inadequate, not just for her, but for her and the baby, the mother's body will prioritize the fetus first. So maternal nutrient stores may be depleted first, and then this leads to deficiencies in the mother's health before the fetus is affected. So these changes reflect a coordinated physiological shift where the maternal body adapts continuously to support the developing life and to take less priority over the developing life that she has. Okay, let's talk about the birth process. Okay, so again, pregnancy ends with the birth process, which is the medical term for this is uh, parturition. So birth approaches. What happens is the levels of progesterone start decreasing, and this allows prostaglandins to start increasing. And what happens is this makes the uterus more sensitive, and it leads to stronger uterine contractions. And at that simultaneously, the cervix starts thinning. It begins to open, and it does this through a process called effacement and dilation. So this is the stretching of the uterus and the cervix, where it's triggered through a positive feedback mechanism. This is one of the few times, or one of the few places where you see positive feedback mechanisms in nature. Positive feedback systems aren't very common, but they are essential for things like this. They're usually essential for things that need a high transformation from one thing to the next. Think of like a caterpillar that's becoming a butterfly. That actually is a positive feedback mechanism. So those kinds of things, big changes in life, are positive feedback mechanisms, and they should be very rare. They shouldn't be very common, or else you get chaos. Anyway, let's keep going back to pregnancy. So you have cervical stretch, right? That stimulates the release of a hormone called oxytocin, sometimes called the bonding hormone, or what do they call it? The love hormone. But that's for that's for psychology. We'll talk about this some other time. And so this intensifies during uterine contractions. As you can imagine, why oxytocin is being released at this particular moment, and it's where there's a bonding that's taking place as oxytocin is being released between the mother and the child, or an increased bonding, not just a bonding. So we end up with stronger contractions. Um, this causes more stretching. This leads to even more oxytocin being released. So you can see the effect of a positive feedback cycle. One thing causes another to increase, increase, increase. It just keeps going and going and going. And so the cycle continues until some major event happens. In this case, it's birth. So once birth happens, that's the trigger event for the positive feedback cycle to stop. So the fetus again is typically positioned in this by this point, the position is head down, facing the cervix, and then during labor is where you have the most powerful uterine contractions at that stage. That's assisted by contraction of the abdominal wall muscles. So when you hear in TV shows and they'll tell the mother to push, this is what they're asking. And so she'll push the fetus through the birth canal, which includes the cervix, the vagina, and the vulva. So after the baby's delivered, contractions continue briefly. This allows the placenta next to be detached and to be expelled. And the final stage is called the afterbirth, right? And so all of this coordinated hormonal muscular events brings pregnancy to its conclusion, meaning the baby is here, time to light up the cigars, and yeah. Okay, this is table 23.5 or 23.5 factors contributing to the labor process. Just as a reminder, focus on these tables since they summarize all of the events of the slides we just discussed, and all of your questions can be answered by understanding these tables backwards and forwards and backwards. Figure 23.25, positive feedback in the birth process. So the positive feedback in birth process. So what we see here in this flow chart, this illustration, is how birth is driven by a positive feedback loop. So we start off where labor begins, uterine contractions, gotta enunciate that a little better. Uterine contractions, they force the fetal head toward the cervix, and this pressure causes the cervix to stretch. The stretching itself activates stretch receptors in the cervical tissue. The receptors trigger a reflex response that leads to the release of oxytocin, and it leads to stronger uterine contractions. Stronger contractions push the fetus farther downward. Again, this increases cervical stretch even more. And so the cycle just keeps going. Stretch, reflex, stronger contractions, repeats again and again. And each step just intensifies the next one. So, which is why labor contractions become stronger, more frequent over time. And the loop continues until the fetus is delivered, at which point the stimulus ends because there's nothing pushing against the cervix or the stretch receptors, right? And so it just ends, and the feedback cycle stops. And so again, this is a classic example of positive feedback, and it's one of the few places uh, in human physiology where amplification, not stabilization, is the goal, right? So we don't want homeostasis here. Um, so the mechanism that we see forms a loop. A reflex is elicited that causes stronger uterine contractions, and due to this, the fetus is moved downward, where the fetal head is forced toward the cervix, and due to this, cervix is stretched, for which the stretch receptors are stimulated. And it just goes on until there's no more um, um, sensation of the cervix being stretched, meaning the baby's already gone through. Okay, here we are looking at figure 23.26. We're looking at an illustration of the stages in birth. So now we're looking at a figure which shows birth as a three-stage process. So each driven by coordinated uterine contractions. So stage one is the dilation stage, uterine contractions. They begin to thin and open the cervix. The amniotic sac can rupture during this stage, and that's when we say that uh, it's the water breaking phase. When you hear on TV, my water's broke. Well, not on TV. You may have heard it in real life. So what happens is this is usually the longest stage of labor, that dilation stage, water breaking stage. Doesn't mean that you have to rush to the hospital right away. Some people do, but not everyone. Stage two is the delivery of the baby, the fetus. So once the cervix is fully dilated, you you have these strong uterine contractions that are assisted by abdominal muscle pushes. So the mother's squeezing her abdominal muscles to push the fetus through the birth canal. Right. So the head emerges first, followed by the shoulders and the rest of the body. So following that is stage three, which is the delivery of the placenta or the afterbirth. And so after the baby's born, uterine contractions, they'll continue for just a little brief period afterwards. And what that does is it causes the placenta to detach from the uterine wall and it causes it to be expelled. So that final stage helps reduce bleeding and it marks the physiological end of pregnancy. So these stages show birth along with the images as a carefully sequenced process. It's driven by muscle contractions, hormones, and most of all, timing. Well, not most of all, alongside timing. All right, let's talk about milk production and secretion. So, production and lactation. So, milk production begins during pregnancy, but it doesn't fully activate until after the birth. Right? So, uh, during the pregnancy, you have the placental estrogens and progesterones that we mentioned that stimulate the development of the mammary glands. And again, the anterior pituitary releases prolactin and something called placental lactogen, and that just helps promote breast development, further breast development. But with these hormones, even though they're present and all this is preparing for milk production and secretion, we don't have it yet. Milk secretion is inhibited during pregnancy. Um, so you'll have these high levels of placental progesterone and placental lactogen that prevent full milk production. But after birth, once the placenta is expelled, those progesterone levels fall. And what this does, it removes that inhibition signal and that allows milk secretion to begin. And so the first, if we're talking about milk secretion and production now, the first milk production or the first milk that's produced is called the colostrum. And this is going to be like a thin, watery fluid, super rich in proteins and antibodies, and it provides immune protection for the newborn. And so the colostrum contains less fat and fewer carbohydrates, so it's not really a lot for energy than the mature milk the mother's going to produce later. But it prepares the baby for the new environment that uh, it'll be in. Um, there's documentaries on this too, and I won't mention them here. I'll mention them in class. What else? Yeah, yeah. So the milk release depends on a neural hormonal reflex. So when an infant suckles, that's going to send a signal, a sensory signal that triggers the release of oxytocin, which you mentioned before. Is that I hate to call it this, but it's a bonding hormone. It's not a bonding hormone, but it's also known to create bonding between mother and infant. So the oxytocin does has like a dual purpose. So this oxytocin causes milk to be ejected from the mammary glands. So suckling stimulates continued release of prolactin that ensures that milk production is maintained throughout the breastfeeding breastfeeding period, right? So if you have an additional effect of prolactin is reproductive. So breastfeeding mothers typically they don't ovulate for several months because prolactin suppresses the secretion of those two hormones I mentioned before, FSH and LH, and I'll just follicle stimulating hormone and luteinizing hormone. And together, these mechanisms they link lactation, infant nutrition, and a temporary reproductive suppression into one like coordinated physiological system. And so the mother can just focus on the baby. This is table 23.6, hormonal control of the mammary glands. This is figure 23.27, mammary glands, non-pregnant and lactating. All right, so mammary glands before and during lactation. So what we see here in the figure compares mammary tissue in two different states. So on the left side, we see a non-pregnant mammary tissue. Did I say that correctly? Non-pregnant mammary tissue. Yeah, so most of what you see here is connective tissue. Relatively small amounts of glandular tissue. The ducts and the glands, they're present, but they're inactive and they're compact. On the right is the lactating mammary tissue, and the difference you can see is dramatic. The glandular tissue is expanded, and the spaces are filled with secretory products, which is milk, and the connective tissue is reduced as the glands enlarge and they dominate the tissue space. Right. So what this shows is functional transformation. The same tissue that was mostly structural becomes highly specialized for milk production and secretion. And what this does, the shift reflects the hormonal changes right after birth, especially the actions of prolactin and oxytocin. These are the physiological actions of specifically oxytocin, even though it also has, like I mentioned before, a psychological effect. And so the highlights show how responsive the mammary glands are to the physiological demands. Okay, we are looking at figures 23.28 and 23.29, milk release from breast. So let's look at the milk release and the what's called the letdown reflex. So what we see here is again a flow chart and an image. And so the figure shows how milk moves from the mammary glands to the infant, and it's a process known as milk ejection or the letdown reflex. And so what happens is milk is produced inside small glandular sacs called alveoli, and surrounding each alveolus are myoepithelial cells, and these play the key role in milk release. So milk flows from the alveoli into the duct system, which leads to the nipple. So the process begins when the nipple or the areola is stimulated by the infant suckling. And what happens is sensory impulses will be triggered. They travel from the breast to the hypothalamus in the brain, and then the hypothalamus signals the posterior pituitary gland to release oxytocin in the bloodstream. And again, oxytocin causes the myoepithelial cells surrounding the alveoli to contract. And so the contraction squeezes milk out of the alveoli through the ducts and out through the nipple. So this is different from milk production itself, which depends on prolactin. Oxytocin doesn't make milk, it helps release milk. Well, no, it doesn't help release milk. Oxytocin releases milk. Right? Prolactin produces milk. What if that prolactin produce? Oxytocin release. Well, if you think prolactin produce, that might help you to distinguish between production and ejection. But what the figures show, or the highlight, is that classical neuroendocrine reflex. You have the sensory input that leads to hormonal output, which produces a physiological response or a physical response. And so the more consistent the stimulation, the more effective the milk ejection. So it's a very tightly coordinated system. It links touch, brain signaling, hormones, and smooth muscle contraction to support infant nutrition. Here we have table 23.7, agents contraindicated during breastfeeding. Okay, so we're talking about our clinical application 23.2, human milk and the benefits and alternatives. So human milk is uniquely suited to meet the needs of a developing infant. It contains about 4.5% fat, about a little over 1% protein. It's a composition that supports rapid brain development. So the higher fat content helps with the formation of myelin. If you remember that from your nervous system chapters, it's a fatty insulation that allows neurons to transmit signals efficiently. So by comparison, cow's milk contains less fat, about 3.5%, and much more protein, around 3.1%, compared to human milk, which is, again, not ideal for human infants' metabolism. So you don't want to feed a baby uh, milk from the store. Um, so the protein in human milk also has this correct balance of essential amino acids needed for human growth. And if you don't remember what amino acids are the building blocks for proteins, right? So it has those amino acids to build all kinds of proteins. So as a result, human milk is just easier to digest than cow's milk, um, and it's just superior to any type of infant formula. There's just no way. Well, I shouldn't say no way. We just can't make anything better. Mother makes everything uh, perfect. So on top of the nutrients, something that's super important is that human milk also provides a strong immune protection, like we talked about in the colostrum. So that colostrum is rich in antibodies, and so lactation, um, as it continues, mature milk supplies additional antibodies, enzymes, white blood cells that help protect the baby against infections. So again, it's like the mother being exposed to the environment, creating the antibodies, and then feeding the baby and providing those antibodies straight to the baby so the baby can be safe and protected. Um, Again, this is something that's just passed down through evolution. So, human milk also contains something called lactoferrin, and this is something that binds iron, so bacteria can't use it to reproduce. Certain bacteria need iron to reproduce. It also contains something called bifidus factor, and that promotes the growth of beneficial lactobacillus bacteria. And that bacteria uh, are the bacteria that produce acids that inhibit harmful microbes in the digestive tract. If you go to HEB and you go to the area where they have the yogurt and the those digestive little drinks, you'll see that in there, lactobacillus bacteria, and it'll tell you like how many it has per per volume. And so you can buy this now. How if they're good or not, I can't tell you. They're not like FDA approved, but you can buy them at HEB, and you can buy them at nutrition stores. Those little tiny drinks. But all of this is included in the breast milk. So again, it's not just what's in the milk. It's not just producing the milk. Again, it all comes back down to timing as well. So feeding patterns also are important to a newborn. So breastfed infants, they typically stop feeding when they're full. Typically, I have seen some chunky babies. Um, Maybe they just weren't full. It took them longer to get full. But this is associated with the if they can stop feeding when they're full, it's associated with the lower risk of obesity compared to bottle-fed infants. So a good reason to breastfeed versus bottle feed. Um, and so, if for some reason breastfeeding isn't possible, then the infant formula is the next best thing. It's the primary alternative. Uh, Formula is based on cow's milk, but it's modified and nutrients are added to it to make it closely resemble the composition of the mother's milk. And again, one takeaway from this particular slide, well, I've been talking about this one for a while, is that human milk, the mother's milk, provides nutritional and immunological uh, substances that are important for developmental progress in the infant. So it's advantageous. It's also safe, um, and it's the best thing compared to the alternatives that exist today. That's all. Okay, let's talk about. We are in section 23.4 in your textbook, where we start talking about aging and the human lifespan. So let's talk about aging and the human lifespan. So this is post postnatal period, right? Which begins at birth and continues until death. And so after birth, both the newborn and the mother they'll both undergo rapidly uh, structural and physiological changes because their bodies are adapting. The baby is adapting to life outside the uterus, but both mother and newborn are adapting to life outside pregnancy itself. So from this point forward, human development becomes this lifelong process of growth, adaptation, and um, unfortunately for you all, aging. Right. So the postnatal period, we break this down into several stages. The um, neonatal period. This is the part that begins weeks after life. So survival adaptation. Again, that's the main challenge, the primary challenge for the infant. Um, well, I should say infant because that's followed by infancy. And so this is a time of rapid growth and neurological development. After that comes the childhood um, period. And this is a period of steady physical growth, increasing cognitive and social abilities. After that comes adolescence, and that brings a lot of hormonal changes, sexual maturation, and the transition into adulthood. Not adulthood yet, but transitioning into adulthood. And that then comes adulthood, and this is characterized by maintenance of structure and function. So um, this is followed by uh, senescence, and this is the gradual decline associated with aging that we see. And after that is dying. And we recognize all of this to be just a natural part of the life cycle, right? What was the thing on the Lion King? the circle of life. And these stages just hopefully remind us that development doesn't end at birth. It continues, it changes, and eventually concludes as part of the same biological story. So we have one, two, three, four, five, six, and seven being the last stage. It reminds me of the play, one of Shakespeare's plays, where they have a soliloquy, and it's the stages of life. And I'll remind you all of that in class. I used to have I used to have the I used to have it memorized. Okay, let's go ahead and talk about the neonatal period. So the neonatal period, again, this spans from birth to the end of the fourth week of life. And so we're going to break these periods down a little further. And so during this short but critical time, the newborn is rapidly adapting to life outside the uterus. The baby begins to breathe independently, well, immediately starts breathing independently, starts obtaining and digesting nutrients, excreting waste, starts regulating its own body temperature, and starts making major cardiovascular adjustments. It starts paying rent, it starts contributing to the bills. I'm just kidding. That comes much later on. Oh, yeah, first breath. First breath is pretty cool. So this is super, it's just cool. So in that first breath, um, it has to be strong enough, one, to inflate the lungs that were, again, these were previously collapsed lungs. And there's a substance that's called surfactant, which we mentioned in respiratory, that helps make this whole thing possible by reducing that surface tension in the lungs. So it allows the lungs to expand more easily. And think of you buy a package of balloons, and some balloons have like a little almost like a talcum powder inside of it to keep the balloons from sticking to each other. So when you blow air into it, the balloon expands nicely. But if you've ever had a pack of balloons you left out for a while, they're in a drawer, and the material sort of sticks to each other, that's analogous to a lung that's collapsed that can't be reinflated. Right. So we have surfactant in the lungs and the alveoli that keeps those things from like sticking to each other, just like those balloons. Um, so again, in the first few days, the newborn relies on stored fat reserves and the colostrum that is getting from the mother for energy. Remember that the colostrum provides the nutrition and the immune protection during this early transition. Also, the kidneys are functioning now, right? This wasn't functioning before for the child, for the baby. So kidney function, it's still immature, right? So it hasn't matured yet. So newborns, they don't produce uh, very concentrated urine. They'll produce very dilute urine, and so their systems for helping maintain like temperature control, water balance, electrolyte regulation, all of this is not very efficient at first. It's still developing. Um, and so everything within this neonatal period, there's a lot of adjustment that the neonate is adapting to. So um, you have multiple body systems that are shifting from placental support to independent function. This is table 23.8, cardiovascular adjustments in the newborn. Okay, we're looking at figure 23.30, cardiovascular changes in newborn. So what we see in the figure is what happens to the circulatory system at that moment of birth. So before birth, fetal circulation relies on um, those special shunts that bypass the lungs and the liver. But once the newborn takes that very first breath, everything changes. So the lungs expand, blood begins to flow to the lungs. Again, the blood flow itself starts increasing because remember we said a little bit does leak into the lungs, and then the oxygen levels rise. And what this does, it causes that foramen ovale, the opening between that right and left atria, it causes it to close. And over time, it becomes a depression called the fossa ovalis. So you've identified the fossa ovalis in the heart anatomy. Before that, it was the foramen ovale, right? It was actually a shunt between the two. So, wow, right? I didn't want to spoil it for you back then, but yeah. And so at that same time, the ductus arteriosus, remember that previously diverted blood away from the lungs, it constricts and it closes, and eventually it forms that ligamentum arteriosum. And so yeah, it's just a ligament there. The other one was the ductus venosus that bypassed the liver during fetal life, so that constricts and becomes the ligamentum venosum. And what this does is it allows blood to now pass fully through the liver. Um, so with the loss of the placental circulation, the umbilical vein closes and becomes the round ligament of the liver. So there are portions of the umbilical arteries that constrict and they form the medial umbilical ligaments. Also, the proximal portions remain functional as the superior vesical arteries that supply the bladder. And what we see here in this figure and what it captures is that complete rerouting of blood flow from placenta-based system to a lung-based independent circulation. So in a matter of minutes to hours, the newborn's cardiovascular system is completely transformed to support life outside the uterus. I imagine like there was an old cartoon called Osmosis Jones, and so if you can imagine a bunch of little engineers and control people, and they're like, we're switching over to, independent circulatory and then close it, blah, 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 whatever. Anyway, that's all. I think this is pretty cool. I think this is a really cool adaptation or change. Okay, let's continue with the next period, infancy. And so infancy spans from the end of the fourth week after birth through the first year of life. And so this is a period, again, of rapid growth. An infant can triple their birth weight, and this will reflect intense physical, neurological development. So teeth actually begin to erupt here, and muscular and nervous systems start maturing. And what this does is starts allowing movements to become increasingly coordinated. And over time, infants start learning how to reach and grasp for objects. They'll learn how to sit and crawl, and eventually they'll learn how to stand. So, yeah, communication begins to emerge at this stage. Infants start to smile, they start to laugh, they'll respond to familiar sounds and voices, and they may even babble or speak a few simple words, baby babble, which I'm fluent in. But they'll do this all by the end of that first year. But because the growth is rapid, infants they have special nutritional needs. So they'll need to have an adequate intake of protein, calcium, vitamin D, iron, vitamin C, all of this stuff that's essential to support bone development, muscle growth, and immune function. So infancy sets the foundation for all later stages of physical, their cognitive, and their social development. Let's talk about the period of childhood. So, childhood extends from 1 year to the age of puberty. And again, growth continues at a very high but steady uh, rate during this particular period or this stage. The primary teeth, they erupt first, and over time they fall out, and then they're replaced by the permanent teeth during this period. And as the jaw and facial bones uh, grow, they'll adapt to the permanent teeth. The nervous system also and the muscular systems, they'll continue to mature. This will lead to full voluntary muscular control. And by this point, children, they're learning to walk, run, climb, and they they're learning how to refine their balance and coordination during this period. Um, Again, also what happens during childhood is bladder and bowel movement. So again, we're talking again from that 1 year on up, but this is very early on. The bladder and bowel control. This is where this part gets established. This reflects the maturation of both the nervous system, regulation, and also learned behaviors. Communication continues to expand, to increase. So children develop their speaking skills, reading, writing, and they start learning abstract thinking during this period. Not masters of abstract thinking, but they start understanding the difference between abstract and concrete thinking. Um, and what this does, it lays the groundwork for learning, problem solving, and so forth. And while all this is happening, let's not forget the effective advancements or the emotional development of children. So children start learning how to regulate emotions. They start learning how to interact socially and how to respond increasingly to complex environments. So the childhood is the period of integration. So physical growth, cognitive development, emotional maturity, all of these are progressing um, together. So in psychology, it's the cognitive, the affective, and the behavioral are all progressing. Let's continue with adolescence. So adolescence spans from puberty to adulthood, and during this stage, the individual becomes reproductively functional as the secondary sex characteristics begin to develop under the initiation of hormonal control. So what happens is that these changes are driven primarily by increases in sex hormones that are released during this time. So you'll have, depending on the hormone, you'll have growth spurts that can occur both in the skeletal and the muscular systems. This will be accompanied by like noticeable increases in appetite to meet those higher energy demands. So um, those of you who've raised teenagers and you see they just eat and eat and eat, yeah, that's where that's coming from because the demand for growth. Under hormonal influences, females will typically mature and reach full physical growth earlier than males, and this is where the saying comes that females mature earlier than men. I'm sorry, females mature earlier than males. Um, uh, that's under the hormonal influences, and so males will typically continue growing for a period much longer. Or what the thing is, females will complete maturation before males. Okay. So some males will continue growing up until like 21 or something like that, where most females will, I think, by 18 they're at their full height. What else? Motor skills. So yeah, motor skills continue to develop. So this means that you have improved coordination, strength, and speed. Again, at the same time, you have intellectual abilities that will mature. So with the development of advanced reasoning, advanced planning, building on the initial abstract thinking. Um, yeah, and then what adolescence ends up being is now a transitional period. Once you complete the adolescence, you transition and you bridge like a rapid biological change with the development of uh, adult physical, cognitive, and emotional capacity. So again, um, physical related to the behavioral and psychology, cognitive is still cognitive, and emotional and psychology is affective, right? Affective traits. So cognitive, affective, behavioral, or physical, cognitive, and emotional. Okay, we are now into adulthood or maturity. So this extends from the end of adolescence through old age. So in this period, physical performance reaches its peak early in the stage. Muscle strength, it typically peaks in the 20s for both sexes. By around age 30, the body begins to lose efficiency gradually. Um, and even though uh, most functions, they remain strong and stable, for many years, it's still decreasing. After 30, around that time, you start developing or you start experiencing uh, degenerative changes, and these things slowly creep in. So, for example, you have sensory acuity that decreases, your ligaments will lose elasticity, and this affects your flexibility and joint stability. So, to put this nicely, when you sit up from a chair, is not as easy as when you were younger. You start getting the aches and pains just for sitting down. And when you get into your 40s, your metabolic rate actually declines. So um, this will lead to weight gain in the 40s. So if you may have been very, very slim growing up, you definitely are in for a shock in your 40s. The hair may begin to start graying out or thinning. The muscles will gradually lose strength. And again, this is for someone who's not, like, say, exercising or maybe not doing strength training or any kind of training on a regular basis. Just naturally, it's going to reduce in strength. The skin will start looking wrinkled. Blood vessels actually start narrowing, and this will affect circulation. So what happens is there is a predictable curve that you can calculate by age of what your expected blood pressure to be, right? And again, you can offset those by exercising and doing things to fight against that. Once you get into the 50s, uh, the changes will become more noticeable. Your near vision will decline. Your nail growth actually slows down. Your taste buds decrease, so you don't taste things the way you used to. The skin is going to continue to lose that elasticity. And around this time is when women typically will stop menstrual cycles. And along with that, muscle mass and body weight will change drastically. So muscle mass will drop, and for women, weight tends to increase. Um, well, mass starts to increase, weight will drop because of muscle. You lose muscle, you lose weight, but mass increases because you're retaining more water and fat deposits. During the 60s, you have mild memory changes. Um, so intellectual functions usually remain sharp, especially with mental and social engagement. So you've seen a lot of those apps where it says like this will keep you sharp for and keep your acuity up. So just basically like you exercise every day to maintain muscle mass, or you jog every day to keep a certain function going. You also have to train and keep the mind sharp and strong by doing puzzles or doing problem solving or something like that. By the 70s, the average height has actually decreased about 1 inch, so you get shorter as you get older. The skin begins to sag even more, and it becomes more noticeable. Connective tissue loss that becomes more apparent. And so adulthood is kind of best understood as this long continuum. As I'm discussing this slide, it's starting to get depressing, but it's this long continuum, right? There's not one sudden decline where it's like at this age you lose this and this and this. It's like it just gradually pulls you down into the abyss. But this gradual physiological change, it's shaped by genetics, your lifestyle, and your environment. So there are things you can do not to stop it, but to slow it down and to enjoy it. Okay, the next section is senescence. And senescence is the process of growing old. So during this stage, right, this is when I guess you can say old, and it won't be an issue. Don't call a young person old. But during the stage, you have the degenerative changes that continue from adulthood, from late adulthood, and the body becomes just progressively less able to cope with physical, and the physiological demands. And so, what these changes reflect is like a long-term effect of wear and tear on the tissues and the organs. You know what? I am going to talk about this slide with a little more enthusiasm because I'm depressing myself as I talk about senescence. So when we talk about certain cell types, right, the cell division tends to slow down, tends to decline, and what this does, it limits the body's ability to repair and replace damaged tissues. So take it easy out there. So I'm going to say, right, don't be doing things you did when you were younger. Um, it's the time to actually enjoy being alive, or you shouldn't have to put too much demand on yourself. The cartilage in your joints, those gradually just wear away, and what ends up happening is it leads to stiffness. You hear older people saying they feel stiff, and that ends up reducing your ability to move around. So you reduce mobility, but you'll have an increase in pain. The immune system starts to weaken, um, and so what this does, it just makes you more vulnerable to infections and diseases. Wow, the experiences you're going to have when you get older with all these infections and diseases, amazing. And I'm trying to spin this in a positive light. Right? It's like, wow, I've never been sick of this before. But what else? Oh, your sensory functions decline. This is pretty cool, right? So your vision and your hearing start dropping. So you don't have to hear all the nonsense that's happening in the world. I think that's probably really cool. Um, and yeah, I think that's probably one of the best ones. And so you're going to have a loss of certain intellectual functions, which is also pretty good because then people won't be asking you to solve all their problems. And it's like, oh, I'm decreased in my intellectual function, so don't ask me to help out anymore. I'm just going to sit here and relax. So senescence sounds pretty dang cool to me if you look at it from the right perspective, right? So what's going to happen is your processing speed is going to be slower. Your memory retrieval will be slower as well, and maybe that's good because then you can retrieve a memory and savor it instead of doing this rapid thing that we do today about like retrieving information and spitting it out. And when you're older, you should have time to reflect and reminisce on memories. No reason to speed up, uh, memory retrieval and data dump. Okay. So what else? You'll Most people retain the ability to have insight, good judgment, and of course their accumulated knowledge, unless they're dealing with a disorder. And so you get to have all that stuff, and you get to experience it and savor it slowly. So it's almost like, from a cognitive perspective, going through the fast food of processing memories and intellectual function of the youth to some finer dining, more of an experience as you get older. So hopefully that helped turn senescence into a positive light for you all. More so for me, since I'm older than y'all, than for you all. And then ultimately, death, like the last bullet point. Oh man, that's a downer. No, I'll turn that into a positive light. So death, right? That results from disturbances to the body systems or from diseases that affect the vital organs that keep you alive. Um, and so death means that you have a, a non-negotiable ending. It's like, yep, this is definitely the end. No wishy-washy. You know that you're done. Your job here is done. And so that's it. So in essence, again, it's not a single event. It's this gradual transition that you can kind of relax and just enjoy the end of your life as you transition into death. Hopefully, I made that as light as possible and as cheerful as possible. This is table 23.9, stages in postnatal development. This is table 23.10, aging related changes. This is table 23.10, continuing aging related changes. Okay, let's talk about another cheerful subject, which is life expectancy. Now, this actually kind of is a cheerful subject. So, human lifespan, we say it has a potential to be about 120 years. Now think about just 100 years ago; it wasn't anywhere near this. So that's kind of a nice thing to think about, especially if you're healthy all the way up until those 120 years. A lot of people don't reach this maximum, right? The majority of people, they'll die early due to illness, injury, system failure, um, rather than reaching that natural biological limit. Or some will die due to other than natural causes, their own competence, in other words, or their own. Uh, yeah, no, I'll just leave it there. But there are countries where, like, the average person is past 80, right? It says most people live past 80. So um, there are individuals who um, pass a certain age uh, at which, like, cancer or cardiovascular disease often occurs, so that they live for a very, very long time, where most people are taken by those kinds of diseases. Cardiovascular is, like, at one point was the number one disease worldwide that took life. So what this means, though, this whole thing is that longevity is influenced not just by your biology, but it's also influenced by your environment, how well you take care of yourself. I mean, as far as like healthcare, medical, and then the lifestyle choices that you choose. So no smoking, no drinking, um, but definitely no smoking. Uh, there's a lot of research that says yes, a glass of wine is good for you, a glass of wine is not good for you, whatever. I think the research actually points to or leans more toward that, like a glass is okay for you or it has benefits for you. Um, but yeah, we should have studied that in the nutrition chapter. So life expectancy, it's a statistical estimate. And again, it represents this projection of how long a person is expected to live based on patterns that have been identified through the study of diseases, which is epidemiology. So these we look at not we, but they look at disease rates, mortality data, and then the population trends. So your book has that the statistic from the in the United States in 2014, life expectancy was about 76.4 years for males, 81.2 years for females. But again, ongoing medical advances are always improving these situations. So, looking at improving public health for one, uh, having better disease management, looking at what actually affects your life expectancy, either here or around the world. All of these are contributing to increasing your life expectancy, or at least understanding what can decrease your expected or your maximum life expectancy. Um, and that's it. So basically, looking at medicine, society, biology, how all that works together across your lifespan. Okay, this is table 23 decimal 11, the 10 leading causes of death in the United States. Um, heart disease, cancer, chronic lower respiratory disease, unintentional injuries, stroke, Alzheimer's disease, diabetes mellitus, influenza, and pneumonia, nephritis, nephrotic syndrome, and nephrosis. And then number 10 is suicide. And again, this is from 2015. Okay, as we're coming to the end of this chapter, let's talk about the end of life. So, when we say the end of life, it's not just like a biological event, it's a deeply personal process. It's shaped by an individual's beliefs, experiences, circumstances. There's a whole area of medicine just for this type of care. And so, that aside, for people who are chronically ill, again, the body often shows recognizable signs as death is approaching or as they're approaching death, however you want to see it. And the signs tend to follow a general sequence. So even though the exact experience varies from person to person, there's a general trend. So the dying process is commonly described in two stages: The first is called the pre-active dying, and this can last up to 3 months. And during this stage, some individuals are aware of what's happening and begin the process of accepting their mortality is, you know, right around the corner. Um, and their interest in like the outside world fades away. They don't really want to interact with the world anymore. Nothing just seems, it doesn't seem worth their energy anymore. Um, so people just lose interest in the news, conversations, friend, and food, and they'll sleep more often. They'll have swallowing difficulties. That's one of their bodies just shutting down. It's no longer needing nutrients or wanting nutrients. So swallowing difficulties can start to develop. The second stage is the active dying stage. This is a little bit more distinct with physical signs. So in this stage, the person may sleep most of the time, um, but they can often, be awakened. You can wake them up. When they wake up, they'll have some confusion. That tends to be common. They can lose awareness of time, and I don't like they'll lose awareness of what time of day it is, whether it's morning or night, or what day of the week it is. And in hospitals, you'll normally hear a nurse go up to a patient, and one of the first things they ask is, can you tell me what day it is? Can you tell me your name when they wake up from a nap? Can you tell me where you are? If there's family or people or visitors in the room, can you tell me who this person is? So, they want to make sure that that confusion hasn't set in. Appetite typically disappears, and then the organ systems, they'll begin to start shutting down slowly. And as the body starts to conserve energy and prepares for death, um, um, yeah, all this is happening for that reason. And then, yeah, it's important to understand these stages because it helps uh, the caregiver, which will be you all, and healthcare professionals. It'll help you all know how to provide the right comfort, provide the patients with the dignity and support that they need to move through these stages. So when we talk about death, um, there's a clinical aspect to providing dignity and support and comfort. Um, so again, it's important to recognize that dying is not a failure of the body, the way the book mentions. It's just the final phase of the human life cycle. Okay, we are in the last section of the chapter of your book, and this is molecular and cellular aging. So the aging process, this is like an overall thing. It's complex and it's kind of difficult to analyze. And one reason is the body functions as this integrated system. So when one particular structure starts to break down, it tends to affect the ability of other structures to function properly. So if I mentioned before that you have one system within the body that has organs that support that system, those organs are also ancillary organs that support a different system as well. So it's not just like one system goes down, the support for the other systems goes down as well. So um, we say that aging rarely occurs in isolation. It sort of spreads out through the system. And the medical field that studies aging is called gerontology. So it might be one of the areas that you might want to work in the hospital. So gerontology is the study of geriatrics. Geriatric is a practice of treating patients in that age, but gerontology examines the biological changes of aging um, at every level of organization. So from whole organs and tissues down to cells and individual molecules. So they look at the whole thing, and they're specialized just in that process. So at the molecular and cellular levels, aging occurs through two broad processes. So there's again active aging and passive aging. And these processes involve changes in the cellular activity. There's damage accumulation that happens over many um, replications of cells, right? And the body ends up decreasing its ability to repair itself over time. So what this does is all of these things help explain why aging affects structure, function, and resilience throughout the lifespan. And what we do is we look at this section to shift our focus on what aging looks like to what's happening beneath the surface and sort of why aging is occurring. All right, let's continue with the passive aging, excuse me, passing. So passive aging again refers to the breakdown of structures and the slowing of body functions over time. And when we look at this at the molecular level, which includes the degeneration of elastin and collagen, these are proteins that provide the strength and flexibility to tissues. Where these proteins start breaking down, skin begins to sag, and muscles start losing their firmness. So passive aging affects uh, genetic material. DNA replication becomes less reliable, and then you have errors in genetic instructions that accumulate. You always have errors in genetic replication, but there's mechanisms to detect those, and so like break any sequence that's about to build something and start over. But as that actually deteriorates, you start passing off, let's say, amino acids and proteins that are not um, that have errors in them, right? And so then that thing gets replicated over and over again, the damaged one. It's sort of like making a Xerox copy, if you all know what Xerox machines are. You make a copy of something, and then instead of making a copy of the original, you make a copy of that copy, and you make a copy of another copy. Every single time you have a little, um, you know, um, something wrong with the image, the next image captures that and then adds to it and adds to it. All right. So, but anyway, so when the damage becomes too great, the cells just stop functioning and they die. So when we look at this at a biochemical level, the lipids start degrading, and the aging membranes and the mitochondria, they begin to leak or they begin to malfunction. And because mitochondria again are responsible for energy production, their decline affects that overall cellular efficiency. Um, I was reading something recently about how creatine actually helps, but don't take my word for it here. I've just been reading a lot of stuff on it. So there are things that they can kind of help, um, not slow down aging, but maybe help one little particular area for energy. So let's keep going. So again, a contributor to cellular aging or cellular damage is the things we call free radicals. You may have heard this before. And free radicals are just molecules that don't have uh, paired electrons. So they're normally if something is paired to each other, they're stable. They're not reactive. But if they're unpaired, I mean, they're kind of like running around. It's like a single person running around a an event for married couples, and that single person is going to be breaking up marriages, right? Versus if everyone's a couple in an event, it's like you're not going to likely have single people running around trying to hit on others and whatever. So a free radical is like a single person looking for a date, and they're always looking for a date, right? And so they're always trying to break something up, and so that makes them unstable. But again, if you want to regain that stability, they have to. If they want to regain stability, they have to like steal electrons from other molecules, and what that ends up doing is it starts damaging uh, other molecules that are stable. So this is not a biochemical rationale to not have single friends if you're a couple, but it does help to have pair up with other couples if you are a couple, right? Or pair up with a if you have a single friend, make sure they're not out there trying to uh, gain your electron. And so what happens is this accumulation starts damaging the molecules. Cells start degrading, so the molecules can't build, a stable cell membranes, and the tissue starts losing its function. So what happens with passive aging is that it reflects what happens when damage gradually outweighs the body's ability to repair itself. And now that I'm talking about this, I do see a nice parallel between this and society. Okay, we're going to continue with active aging. I won't spend too much time on the asides and stories. Let me just get through this quickly. And so, what activation involves is new cellular activities or the appearance of new substances rather than like simply things breaking down. And a good example is the formation of something called lipofuscin granules. So, these are pigment like accumulations and they result from the breakdown of lipids, which things that are happening inside the cells, aging cells. So, over time, a lipofuscin builds up, um, especially in cells that are really long lived like neurons and muscle cells. And so this can also involve changes like in the immune function with active aging, and in some cases like autoimmunity develops, meaning that immune systems begin to attack the body's own cells, and this also contributes to tissue damage. All of this stuff happening. What else? Oh, apoptosis, right? So something else that happens is programmed cell death, which is the medical term is apoptosis. And again, this is not like an accident. This is a controlled, genetically regulated process. And again, this process begins even before birth and continues throughout your entire life. So, for example, fetal brain development, about half of newly formed neurons, they'll undergo apoptosis, while those that kind of establish very strong synaptic connections to survive, they'll continue. So you have apoptosis and mitosis, which are opposite of each other, but they're complementary processes. So mitosis will create new cells. Apoptosis removes just removes cells, right? Cells that are no longer needed or damaged or whatever. And so this apoptosis mitosis cycle sort of maintains a balance in tissues. But active aging reminds us that cell death, again, is not failure. It's just a normal part of life. So essential for development, maintenance, and healthy aging. Just a normal part of life. And we are on clinical application 23.3. So this is living to 100 and beyond. So there are people who live past the age of 100, and we call those people uh, centenarians. And there's research just on centenarians because everybody wants to know what it takes to live that long. And so, what the research shows is that environmental factors like lifestyle, nutrition, and healthcare, all of these play a major role in the death between ages of 60 and 85. But beyond that, it's where genetics becomes more of an important role in determining longevity. So, centenarians tend to share some characteristics. Like many have high levels of HDL, that high density lipoprotein cholesterol, and that's kind of super important for developing strong cell membranes, right? And so again, this appears to significantly reduce cardiovascular risk, and it can possibly add up to 20 years of life. And someone who maintains a normal to low body weight, they've never smoked, um, if they learn how to manage stress effectively, they're not always hypertensive and agitated and anxious. All of this shows a low incidence of dementia as well. So when we look at centenarians genetically, uh, it seems like they inherit some of these protective gene variants. So um, they can carry normal versions of genes that, when mutated, cause disease in other individuals, but the genes they have most strongly associated with like exceptional longevity influence things like glucose metabolism and insulin secretion. And again, that other than keeping you from becoming diabetic, it maintains a normal, healthy weight. Having a good um, or strong immune system function, having a good control of the cell cycle, the mitosis, apoptosis, maintaining, and again, through nutrition, hopefully, a strong level of high HDL, high density lipoprotein cholesterol, and then the breakdown of low density lipoprotein cholesterol. You don't want too much of that. Then you can have what's called cellular response to stress. And when we say cellular response to stress, we don't mean like you have an exam tomorrow and you have a migraine. You can picture someone with their hand on their temple because they're stressed. I'm talking about stress that actually affects the like cellular respiration and so forth. And then the production of antioxidant enzymes. Right? And so, what these factors suggest is that long life is just not the result of like a single gene or a single behavior. It's a combination of quite a bit of things that we can sort of like actually put down to paper. So, genetics, resilience, and lifestyle factors. So, these are some things that I have a problem with that word. Centenarians, right, have good control over, right? And allows them to have good adaptation, protection throughout their life, and it helps them balance their stress at the molecular level. All right, so that's it for this chapter. I want to thank you for your time, and that's it. I'll see you on the next lesson, or I'll see you on the other side.